We have Dr. Marcelo Freire, who is from the J. Craig Venter Institute at UC San Diego and is an associate professor there in the Department of Medicine and Infectious Disease. His talk title today is Understanding Salivary Proteomic Signals in Health and Disease.
Thank you for the introduction. Thank you, Brittany, for the invite, and all of you for attending. So first of all, I want everybody to log in and go and use the salivaryproteome.org, which is a free resource. It's a database that was actually not created by us at JCVI. We currently manage this database and we develop new tools. But it was developed at NIDCR before, and one of the speakers here, Dr. David Wong, was one of the pioneers with others that actually started this concept of thinking proteomics database. So we will talk about what we're doing now and what's our vision for salivary proteome. I'm a faculty here at the Craig Venter Institute in La Jolla, and also I'm a faculty in the UCSD. And the JCVI is merging actually with UCSD in many aspects. We are actually officially part of UCSD institutes. So if you don't know, it's a building that's LEED certified. It's one of the only lab facilities that has LEED certification, founded in 1992, and with the goal of understanding genomics and some other fields of fundamental understanding of life. Just highlighting a couple of things that happened here in this building, like the pioneer paper in the genomic sequence and the subsequent global impact, including sequencing of Drosophila, the dog genome, and the human genome. And also one of the seminal papers for microbiome, when in 2006, our previous leader, Karen Nelson, investigating 2 gut samples and did a shotgun analysis of the gut microbiome of these 2 samples, which allowed the field to explode, the microbiome field to become a phenomenon for us to explore and start to understand. So what is the question of my lab? When I see this map here, and that this map comes from the CDC, and it changed color here from blue to red, it's not exactly a representation of politics. I just want to make sure that you know I'm talking about science. But the idea here, as it's changing the colors, it's showing that there is increase of chronic conditions. So chronic conditions here included are obesity, type 2 diabetes, and many others. But the point I'm trying to make here is there is a tendency through time, and this tendency continues to rise. And chronic conditions different than infectious diseases. They don't have a one gene, one agent, or they are not exactly conditions that we could, in fact, have a vaccine at this moment for it. So chronic conditions, they have something in common. For example, periodontal inflammation, which is a disease in the jaws, will have chronic inflammatory signals. Type 2 diabetes, uncontrolled type 2 diabetes, we also have very similar signals of inflammation. But when you treat each disease in chronic inflammation, we'll only focus on certain targets. For example, in type 2 diabetes, people will focus mostly on sugar control, but not on the inflammatory molecular signals. The same thing for periodontal disease. Most practitioners will focus on control of the microbes, but not exactly targeting inflammation. So it's a passion of mine, and it combines some work that was done by Craig Venter with the genome and also the microbiome, because those 2 are communicating, and those signals are very important to my lab. So molecules are driving pathogenesis, and molecules can be detected before disease happened. So our model in medicine is very much a model of let's diagnose a disease versus let's monitor your health and understand the molecules that are driving pathogenesis. So as you know, the mouth is an organ that's complex. There's a tongue, there's niches in the palate, salivary glands, there's a saliva here, gingival tissues, salivary ducts. So a lot of the sites harbor environments that favor microbial growth. And this is niche specific. We'll have microbes growing in the tongue, specific microbes in the palate, specific microbes in the gingival crevice, in the clavicular fluid, and specific microbes will be found in saliva. More variability in saliva, but a lot of it is actually very niche specific, and even if you don't get the species, they're very much the same, you will get functions, they're conserved, and you will see similarities of functions happening in the gingival fluid versus in the tongue, or versus in the hard palate. So we have several niches in the mouth that are quite interesting. Here's just a photo of the initial human microbiome project demonstrating diversity of the oral versus gut and some other sites of the body. In this case, they utilize dental plaques. So what is it? What is the composition of saliva? Saliva has a lot of epithelial cells, a lot of immune cells. In fact, when I started this field many years ago of looking at immune cells, you know, many people in the audience would say, okay, we don't have immune cells. They're alive in saliva. Okay, so I don't need any graph, but I need a video to show that actually saliva has a lot of immune cells. And if you place a chemoattractant, these cells will actually go in and out. And they can do that in blood vessels as well. So there are macrophages, there are neutrophils, and many others. And when you try to do immune landscape, evaluating the composition of saliva, you see that there's a lot of neutrophils and monocytes, and you can characterize the variability and the type of cells. In fact, we start to map what type of neutrophils those are, and ask the question, is this just one type, or there are subtypes? And we can do this machine learning program called DAFI and investigate each of these populations are based by their shape and size. And by doing this, we can in fact investigate the types of population that are there. And then after that, investigate the function. But other ways to investigate saliva, it's collecting the fluid and evaluating the fluid by proteomics. So here is the database of proteomics. It's a free resource, and please go and let us know. We are working on a version 2 right now. And this is a salivary initial plot. It's not a perfect plot, but start to dissect the whole saliva, which is a passive drool, from isolated saliva from glands, parotid glands and submandibular sublingual glands, versus blood plasma. In this case, blood plasma samples are from human plasma consortium from the peptide atlas. But the idea that we have common proteins between saliva and blood, and it would be very cool to, in fact, look in health and disease and understand if we could monitor blood molecules in saliva more reliably. But also for the biologists that are interested in isolated glands, they can look into their specific molecules that are of interest, for example, for diseases like Sjogren's syndrome or oral cancer, and so on. So we had this paper that we used to release this version of the wiki. The wiki is currently a cloud-based project, and JCVI is doing the informatics aspects of this project. So this is just a graph here showing the difference between oral cancer versus healthy controls, and whatever is upregulated and downregulated. Because we now not only have these data sets from isolated glands from healthy subjects, but we also have health and disease data sets that we can evaluate. So this is open to anyone. Just because of time, I'll go a little quicker here so I can go through some of the COVID data sets. But the idea is that you can apply your data set or a gel from salivary proteome, and you can start to ask questions that identify. These are all generated by the wiki, which is the Human Salivary Proteome database. Okay, so how are these molecules driving pathogenesis and how does this really happens? Here's a study model where we collected saliva and plasma from in the beginning of the pandemics, when we started to have people exposed, but then going through convalescent phase. And we started to investigate and ask the question. So it was more a convenient cohort. Where we were fascinated was to continue to look at the immune response, continue to look at inflammatory signals. And we started to evaluate our raw data. Here's a plasma of COVID groups versus healthy controls, and here's saliva. And we started to see some difference in the clusters, not exactly significant difference when we look at the entire analysis. But then I will show you what we've been doing to understand our data sets better. As you know, even today, there are many new methods for proteomics, for saliva and plasma, preparation systems to really remove the most populated cells. For example, in plasma, albumin, it's difficult to do reliably, so we can still have the raw data, the raw sample, and then we can investigate all the other proteins that are present in the fluid. So just to highlight here, one of the actual increase that we found in plasma was the fact that we had a lot of this fibrinogen increase. And we know that fibrinogen is very much involved in vascular damage. An excessive amount of fibrinogen is involved with neutrophil extracellular traps. And these neutrophils, these innate immune cells that I showed you before, that are present in saliva, they in fact form these net-like structures, and they influence some other systems in terms of damaging the vascular tissues and creating some more of a fibrosis in the tissue. So we did machine learning to evaluate our plasma and saliva, and we started to dissect. Each of these dots here represent an increase or predictability that those are increasing disease, and we start to see fibrinogen and some complement functions. And in saliva, we actually also found fibrinogen, but here we see a calcium channel molecule that's quite important for, it's an early marker for gene expression in innate immune cells. We all like those graphs and machine learning analysis, but there's something really valuable to go through the lists, of your molecules and evaluate what is up, what is down, and what's the biological meaning. So we learned a lot. We learned that innate immune response of the mucosa are quite affected. So it's not just about antibodies. Infectious disease such as COVID or long COVID, the chronic version, it's not just looking at antibody levels to the virus, but actually there's way more to look into. For example, neutrophil markers called myeloperoxidase, which are markers that are involved into neutrophil phenotype. And if you do have excess amount of myeloperoxidase, you do have a high destruction of the tissue. So it's not uncommon to see increased myeloperoxidase in patients with periodontal disease, for example. But here we're talking about COVID, and patients don't have periodontal disease. But in fact, salivary neutrophils are expressing a high amount of MPOs and lysozyme Cs, which are quite important for relevant for tissue degradation. So the wiki is evolving, and we are doing a version 2, or wiki 2.0. Here's a version of the analysis panel. We will find ways to improve how we evaluate proteomics. Every paper has a problem of how we analyze. So it's very hard to put together a metadata paper and compare, because everything was done so differently. For example, the filtering aspect. Some investigators in the biomarker field, like the proteome from NCI, from the cancer groups, they will exclude any protein that it's present in the samples less than 30%. So the frequency matters for filtering. But then if you're using infectious disease, if some proteins are only shown on the disease groups, even if it's a low frequency, it does matter to keep those. So each field has a different filtering system. They also have a different normalization strategy, whether they normalize by the sum or by the median, or they do, a few versions of the sample normalization, like log transform, or they account for health versus control. So these need to be taken into consideration when you're putting together these data sets. Right now, we have more than 800 data sets in the wiki, and we are working on this and testing to see what's the best way to do across the board. Our page, we have also an explore page where you'll be able to look by biofluid, you'll be able to investigate by frequency of proteins and understand the signature types, also by year of the study. So you'll be able to upload your data, download the data readily for your studies. One example here is that our previous data that we just showed, that we have healthy versus COVID being red and healthy blue, you start to have one level of separation. But then as you learn different types of analysis, like the Wald test or the Wilcoxon rank sum test, you have a different separation, and you can start to appreciate the difference or the top hits that you could later validate in the lab and run your ELISAs or run immunoassays to investigate if those markers are indeed relevant. We continue to see through these iterations of separation, we continue to see those calcium channels and some other markers that are involved with neutrophil behavior, such as myeloperoxidase and some others. These methods are being developed right now. Here's one level of the pipeline. This analytical pipeline takes in consideration predictive modeling, pathway enrichment, correlation of the relationship of the samples. And then our goal is that the user, we have a very clean output for biomarker, for example, identification, where they can get a heat map and they can get just regular box plots to understand the difference between specific markers or across the data sets using volcanoplots. So it will be a metadata-driven analytics interfacing the platform that's web-based, but also allowing people to explore and analyze salivary proteins. How am I doing for time?
I was actually just going to pop in and let you know that you've got about a minute left if you want to wrap up, and then maybe if we've got time at the end of the webinar, people can ask you questions then.
Okay, awesome. So I'm finishing here too. So the idea is this: I'm doing a lot of the immune response. We have a relationship with the Foresight Institute that we are doing also the microbiome response. So combining those networks data allow us to develop the networks of host microbial signals that are present in proteomics. We are considering the metadata, the clinical information, and also the molecular data. But the idea of this community knowledge base is to evaluate health versus disease, but also get to a point where we're doing early markers detection, but also immune detection, so we can identify markers in the early stage. So please use salivary proteome. Here's the URL again. And I'd like to acknowledge my collaborators and the funding source from the Conrad Krebs, Cohen Foundation, PolyBio, and IDCR.
So with that, happy to turn it over to Dr. Ellen Kersh, who is Chief of the STD Laboratory Reference and Research Branch at the CDC, the Division of STD Prevention. And her talk title today is A Public Health Need for Bacterial STD Self-Testing or Specimen Self-Collection Options. Dr. Kersh, over to you.
So thank you for the invitation. That is wonderful. I will be talking about something quite different, a public health need for bacterial sexually transmitted disease, self-testing or specimen self-collection options. Let's see. I have no disclosures in these findings, and opinions are mine alone. So rates of reported chlamydia and gonorrhea cases are stubbornly high. In 2021, the chlamydia rate among women was 630 per 100,000, and the gonorrhea rate was highest in men. It was 250 per 100,000. And before COVID, chlamydia and gonorrhea were the #1 and the #2 most frequently reported nationally notifiable condition for CDC to worry about in the United States. So there is a great volume of tests, diagnostic tests, run annually in the United States that result in these numbers. So for chlamydia, we estimate 1.6 million, and for gonorrhea, 0.7 million. We only get reports of positive cases, so that means we don't know exactly how many tests are actually run, but we estimate it's at least 19 million, which is a substantial amount. Much of this is due to CDC and other agencies' recommendations for screening, so asymptomatic screening and also diagnostic testing. Most of these tests are done by dual NAT, nucleic acid amplification test, for both gonorrhea and chlamydia together, and the most common specimen type is urine or vaginal swab for women. But there are also tests for other sites, like the oral site or rectal sites, depending on reported sexual behaviors. And during COVID, many providers explored contactless testing services through telehealth services and also testing. And this is what the focus of my talk today is. So before I get into that, there are some definitions. So of course, we're all familiar with home self-tests that would be depicted on the left here, and these do not exist for gonorrhea and chlamydia currently. So we do not have a standalone self-test. And for that reason, there is a model that is depicted on the right here, where there is a person at home that can either interact with a clinic or with a commercial site and order a test or be prescribed a test, where they would collect a specimen at home that then gets sent to a laboratory where the test is performed, and the result is then sent back one way or another to this person. So when we talk about self-collection mail-in testing, that is the model we mean that is very prevalent currently. And home in this context is just easily understood. Everyone knows what that means, but it is not intended to exclude the homeless or incarcerated or other persons who may not be testing at home. So the landscape of this sort of testing after the pandemic is as follows. So many STD clinics or other clinicians or other sexual health service providers are offering this either direct to consumer or as telehealth service. The public has increasing awareness and familiarity through COVID self-testing with this type of model. And there is very strong public interest and demand for various reasons that I would like to get into in the next slides. So there is scientific evidence and also an existing public health recommendation by our colleagues at the World Health Organization that this model makes sense for both HIV but also STD, sexually transmitted disease, testing such as chlamydia and gonorrhea. So our colleagues actually have guidelines that are depicted here for so-called self-care interventions for health. And it's no accident that these were first issued for sexual health issues such as the topic we're talking about. It is considered a people-centered approach that enables active participation in one's own health. It can reduce barriers to access to healthcare. And the goal is to support and complement existing approaches to reach people who may not otherwise test. It's very important to us to reach more people. And the guiding principle is to both benefit the individual that gets tested with their health, but also improve health outcomes at the population level and drive down rates. So our colleagues at WHO did a systematic literature review of both behavioral research and cost effectiveness, and also existing randomized clinical trials on the performance and uptake of such testing. And just to summarize their whole body of work, we believe this is they had a wonderful summary of the advantages and also some problems with this type of testing. So on the plus side, it does empower individuals to take measures to protect themselves and their loved ones. The self-testing or home specimen collection has very high acceptability. It's actually preferred by many people. And that is because it's convenient, it's speedy, and there are no transportation barriers. Sexually active people are typically young and active and don't have much time to go to see a doctor. Very important for STI, it allows avoidance of stigma, embarrassment, and personal questions that you are exposed to if you seek healthcare. And it does allow self-control of any consequences of a diagnosis. And it does, very importantly, reach people who distrust or avoid the healthcare system altogether. On the negative side, one has to be careful to provide linkage to care so people can actually get treated. And we're also worried that some tests may have lower sensitivity and some other cons that I won't go into here. So we at CDC are currently doing our own systematic literature reviews for effectiveness here in the United States in our settings. And very briefly, in the interest of time, won't go through this in detail, just would like to say that in clinical trials that are relevant for the United States, we also find that home self-collection or self-testing, when offered, compared to clinic collection, it does seem to increase the uptake of testing here, separated for women and men up to 6-fold for men, and a smaller effect for women, but still a sizable, acknowledging heterogeneity in these trials, but still trending in the direction that we're interested in, which is an increase in testing. We do not find problems with linkage to care. Folks who are testing at home or want self-collection are usually motivated to seek care, as is reflected in these data. And the test performance, comparing one person that tests either at a clinic or at home, is also quite impressive. High specificity. There can be some reduction in sensitivity, but not in all studies, and it can be mitigated. So this brings me to this topic. So currently, we do not have any FDA-cleared tests that are cleared for home specimen self-collection. Yet this is very ubiquitously offered. Just in preparation for this talk, just 2 days ago, I went on Amazon to search what comes up. So when I put in SDI test kit, it auto-completed, because this is apparently a search often done. It completed to at home for women, and this was a screenshot of what came up very quickly. And you can see here that it's offered ubiquitously. There are many offerers. As you can see, there's 3 on screenshot here, and they're randomly selected just what came up. So it's not an endorsement or any statement of the quality of these tests. And you can also see the prices. So this is considered a fairly expensive model. These are out-of-pocket costs that not everyone can afford. And again, these tests are not FDA cleared, although the FDA is currently apparently not enforcing their use. So here's a table in general about what might be the problem why we haven't seen FDA clearances yet. So gonorrhea, chlamydia, and nucleic acid tests are offered by many companies, many brands, and they typically have many claims for different specimens. So most have physician-collected vaginal swab. This was kind of the original test. And then also urine as a specimen. Of course, that's always patient-collected, but under physician supervision. That's how they're approved. And later, there were additions of additional swab specimens, so penile, meatal, rectal, throat, lesion swabs. These are all approved as physician-collected in a clinical setting. We know that there is some gray zone if that some of these swabs get collected in physicians' offices after physician supervision, sometimes as lab-developed tests, sometimes just that's how it happens in practice. However, that's not how they're approved. And then on the right side of this table here, very importantly, none of these tests as of today have a claim for patient-collected non-clinical setting, meaning at home, yet that's how they're used. Here are some additional challenges. So we do have many test systems that offer these millions of tests, and I've randomly selected a few here on the right. So many of the big companies offer such a test, and all of them, or should I say most of them, have their own collection device depicted here. So that is a proprietary system with a transport media where the swab gets deposited in for stabilization, and then it has to go on the same test system, and these are not interchangeable. So in order to get new claims for self-collection at home, each company would have to get a new clinical trial and a claim done for their test system to be tested for safety, and then also test accuracy after transport. This is, of course, expensive and is a big hurdle to climb. And we are hearing from many manufacturers that are not immediately planning to do that due to the expense. So here are some future potential solutions. And of course, the experts at companies such as yours or organizations such as yours are invited and encouraged to think about this problem and also offer solutions or get active in this area, which is why I'm speaking here today. We're really hoping that we will get some interest in the diagnostic landscape, how this problem could be solved. So for mail-in gonorrhea, chlamydia testing options, of course, the manufacturers could obtain their own claims, which many of them, again, have already indicated they may not be doing due to the expense. Then we are aware at CDC that some telehealth service offerers are working on obtaining claims from FDA for their service model. And that's wonderful. We're definitely happy that they're doing that. And we're also somewhat concerned that this would be expensive and that would also be difficult to incorporate into existing public health service models where, you have the clinic, you have the lab on site. So using this whole service elsewhere would be difficult to incorporate. So we are hoping that there might eventually be funding or an interest in doing public health-led clinical trials to evaluate the specimen transport maybe in a more universal way, like a dry swab that would not always rely on proprietary transport media for each company. And such a dry swab specimen could then perhaps be accepted for multiple tests once they arrive in the laboratory. So we are aware that there was similar work on saliva as an accepted specimen for COVID tests under emergency use authorization. And that was also a reason why we thought it would be a good idea to speak with you all today. In case you have any ideas how this could also be done for our problem here. And then, of course, the development of true self-tests, standalone tests at home would also be a solution that unfortunately is not immediately on the horizon as far as we are aware. So this is just another depiction of what I'm talking about. Hopefully there could be discussion or the development of a strategy to conduct clinical trials where these dry swabs could be collected and then be acceptable in various test systems that are already FDA cleared. So in summary, and this is my second to last slide, I hope I was able to convince you that there is a need for chlamydia, gonorrhea, self-testing or specimen mail-in test options for the home. We think that specimen transport in a dry swab format warrants further evaluation and could be a cost-effective solution to current obstacles. And overall, increasing the uptake, speed, and affordability of testing by removing barriers is a strategy that we are pursuing to reduce STI cases, which remain unacceptably high. Thank you for your attention. And I would like to thank Amanda Smith and Phoebe Thorpe, who are currently doing our systematic literature reviews for this project. Thank you.
Thank you so much for that presentation. We have time for a question if anyone wants to either use the raised hand feature or post one over into the chat. And I do have one to kick us off perhaps, and that's you'd mentioned about inviting people who are part of this network to collaborate or get involved or offer feedback. What's the best way for them to do that? Is that emailing you directly, or is there a website they should go to?
Emailing would be great.
Fantastic. Would you mind adding your email address in the chat, and I can make sure that it gets sent out to our attendees as well?
I will do that.
Thank
Yes.
you.